હવે સમજો, હવે અત્યારે તમારે કોઈ બી પર્સન એમ કે મારું જે નામ છે ડેનિસ કનેરિયા ઉપર સૌથી ઉપર દેખાશે તમને, એના ઉપર મોર માં જઈ અને રિપ્લેસ સ્પોટલાઇટ કરી, એટલે હું જ્યારે બોલતો હોઈશ ત્યારે મારી સ્ક્રીન ઉપર જ દેખાશે.
સ્પોટલાઇટ ફોર એવરીવન સ્ટોપ વિડિયો રિપ્લેસ વાળું નથી આવતું ઓપ્શન.
હા, એટલે મારો
તમારામાં નહીં આવે,
મારા નામમાં આવશે. મારું જે ડેનિસ કનેરિયા લખેલું છે ને, ડેનિસ કનેરિયા, એની ઉપર તમે ક્લિક કરો, મોરમાં જાઓ અને રિપ્લેસ પાર્ટિસિપન્ટ્સનો વિડિયો ઓફ હશે ને એના નામની સામે સ્પોટલાઇટ નહીં થાય. એટલે તમારે કમ્પલસરી જ્યારે બી સ્પોટલાઇટ કરવું હોય ત્યારે સામેવાળા પાર્ટિસિપન્ટ્સનો વિડિયો કમ્પલસરી ઓન જોઈશે.
હા,
મેં તમને YouTube લાઈવ સ્ટ્રીમિંગ માટેની લિંક તમારા ચેટ બોર્ડમાં મૂકેલી છે અને WhatsApp બી કરેલી છે. એ લિંક તમે તમારા પાર્ટિસિપન્ટ્સને મેલ કરી શકો છો અત્યારે.
સારું
કે ભાઈ આ લિંક થ્રુ તમને ડાયરેક્ટ લાઈવ સ્ટ્રીમિંગ જોવા મળે.
ડેનિશભાઈ મેં નાનકડું એક ગ્રુપ બનાવ્યું છે અમારા 5 જણનું.
હા, એમાં બી નાખી દીધું છે. એમાં
નાખ્યો ને તમે?
નાખી દીધું છે.
હા, અહીંયા મૂકી દો ને. હા, સારું, એ જ બરોબર છે. તો અહીંયા મૂકી દઈએ ચેટમાં.
અમે
ચેટમાં મૂકો, અથવા તો જે પાર્ટિસિપન્ટ અહીંયા જોઈન થશે એને ચેટની જરૂર નથી. કે કૃપાબેન કહેતા હતા કે અમુક લોકો જે જોઈન નથી થઈ શકવાના એ લોકોને મેલ કરી દો આ લિંક અત્યારે.
અરે એટલું અત્યારે કૃપા ઓલરેડી અત્યારે ગભરાયેલી છે. કૃપાબેન તમે યુ ડોન્ટ વરી.
ના, હું મેલ ચાલુ કરું છું અત્યારે. જે ટાઈમ છે 11 વાગ્યા સુધી, જેટલા મેલ થાય એટલા કરીએ અને મેડમ WhatsApp ના ગ્રુપમાં સ્પ્રેડ કરશે. જેટલા થાય એટલા થાય.
અરે અહીંયા જ મૂકી દેવાનું, જેને ત્યાંથી જોવું હોય ત્યાંથી જોવે, પણ ચોખ્ખું લખવાનું કે તમે ત્યાંથી જોશો તો તમારું પાર્ટિસિપન્ટમાં નામ નહીં આવે. એટલે જ્યાં સુધી બને ત્યાં સુધી આ ઝૂમ મીટિંગ જ અટેન્ડ કરો.
ઝૂમ માંથી એ લોકો જોશે, ભલે ત્યાંથી જોવે મેડમ, વાંધો નહીં. એ લોકો ફીડબેક ફોર્મ ભરશે ને એના પરથી એ લોકોના સર્ટિફિકેટ બનશે. ધેટ્સ સો ફાઇન.
મેડમ એની વાત નથી. ટુ ગેટ અ લિસ્ટ ઓફ પાર્ટિસિપન્ટ્સ we need them here, otherwise on YouTube we can't see who is watching.
હા, બટ 300 તો થઈ જશે. એનથી વધારે થશે ને એ લોકોને જ
ઇનકન્વીનિયન્સ છે કે 300 થાય ત્યારે ને તમે જોજો કે 300 થાય છે. ત્યાં સુધી નથી.
એ 300 લાસ્ટ ટાઈમની મીટિંગમાં બી એવું જ હતું. મેડમને 600 પાર્ટિસિપન્ટની એન્ટ્રી હતી પણ 300 નતા થયા. 270 પાર્ટિસિપન્ટ્સ.
અચ્છા, નહીં, અમારે 1100 રજીસ્ટ્રેશન હતું ને ત્યારે 722 થયા હતા. 1100 માંથી 500 જેટલા નતા આવ્યા. એ તો વાત સાચી છે, પણ
મેડમ હવે બહુ એ વાત જૂની થઈ ગઈ. હવે શું છે કે દરરોજના 5 વેબિનાર લોકો એટેન્ડ કરતા હોય. કોઈ અમારે 800 850 જણાએ કર્યું તો એમાંથી 250 જણા આવેલા. ડેનિશભાઈને ખબર છે હમણાં. એટલે ડોન્ટ વરી. હવે તમે ખાલી ટર્ન બાય ટર્ન એક પછી એક બધાનું પેલા પ્રોગ્રામ પ્રમાણે સ્પોટલાઇટ કરીને પ્રેક્ટિસ કરી લો. પછી બધાને એન્ટર કરી દઈએ 10:55.
ઓકે.
પ્રીતિબેનને જોઈન કર્યું હોત ને તો પ્રેક્ટિસ થઈ જાત અહીંયા.
હા હા, એમને કહું.
ના, તમે પહેલા બીજાની આગળની પ્રેક્ટિસ કરી લો, બીજા બધાને. છાબરિયા સર આયા છે? મેડમ કૃપાબેન, unmute yourself.
હા, બોલો મેડમ.
Madam, if you are confused about spotlighting, highlighting, I can do it, but there is no confusion. Na તમે જેવું તમે તમારું pointer એ નામ પર. Now you must know our names. See, I know all your names. હવે તમે કહો કે જૈન સર નથી, ભાનુકુમાર જૈન is the Jain sir. Okay? So you must know all this. એટલા માટે અત્યારે તમને અમારા ત્રણના મોઢા ખબર પડી ગઈ.
એ તો બધા ખબર. આ ચારે સાકેત સર એકવાર કર્યું, ઝંખના મેડમ કર્યું, એકવાર જેનિશ સર, બધા સરનું એક વાર માં
અને પ્રેયર આટલું કરી લો. ચિંતા નહીં. હવે બિલકુલ શાંત. હવે કશો જ વાંધો નથી. ઓકે, હવે મેડમ બધાને એડમિટ કરી દઈએ. ડેનિશભાઈને જવું હોય જવા દો. બરોબર મેડમ, એડમિટ કરી દો બધાને. અને મેડમ કીપ વોચિંગ યોર ચેટ. આઈ મે રાઇટ ટુ યુ સમથિંગ. ઓકે
ઓકે.
આપણે ઇન્ટરનલ ચેટ થી વાત કરશું. હવે બધાને એડ કરી દો. 10:50 થઈ ગઈ મેડમ, આવવા દો બધાને. ડેનિશભાઈ તમે કેટલું રોકાઈ શકો એમ?
Good morning, Jain sir.
Good morning, Chabaria sir.
Good morning to all.
Good morning, Chabaria sir.
Good morning, Dr. Chabaria sir.
Good morning.
Oh,
Good morning,
Chabaria
Chabaria.
sir. Hello, hello. Others, we request all others to please keep their video off and audio muted. This is only for our testing. Please keep your video off at all the time. Yamini. Thank you. Achrekar sir, Charekar sir, please just speak something and check.
Hello, good morning, madam. I hope I am audible and visible as well.
Perfect, perfect.
Thank you.
Very clear.
Very clear.
Thank you, sir. Welcome, sir. I'm Alpana.
Nice to meet you, madam. Good morning.
Nice to meet you too. And our principal, sir, Dr. B.K. Jain, sir. Bhanukumar Jain.
Good morning, sir.
Good morning, sir. Good morning.
So, we'll start very soon. I think Chabaria sir is also here.
Yeah, good morning.
Good morning, Chabaria sir. So I think, sir, why don't one of you call Kalro sir and tell him to join us?
Yeah,
Then we
just
can start.
sure.
And Mamta madam, where is she?
Ma'am is on the way.
Okay, no problem, no problem. As soon as she comes, then we can start.
Sure.
So meanwhile, I request all other participants who are not going to speak today, kindly keep your video off and kindly keep yourselves muted all the time. Thank you very much.
Kaldro sir has already joined.
Very good.
Good morning, everybody.
Good morning,
Good morning, sir.
Good morning, sir. Good morning.
Good morning, sir.
Akshay sir, this is Professor Kalro. He's our academic advisor.
Very good morning, sir.
Kalro sir, Akshay sir is our first speaker for the day. And our second speaker, somebody on his behalf has already joined. And sir, I think will join later.
Yeah,
Good morning, Preeti madam.
Good morning, ma'am.
Good to see you.
Yeah, ma'am. Same here.
Mamta Ben is here. So I think we can start in 5 minutes.
Okay. Only 60 participants have joined yet, ma'am. We have 807.
My gosh.
Yeah.
I just had a small request. If you could make Dr. Saikat Malik from our laboratory the co-host, because he's going to answer the Q&A. So if he's the co-host, you can see the Q&A section.
Okay. Okay. Saikat Malik, I'll find out.
He's in the meeting right now. Just request you to make him the co-host.
Just make him
already co-host, sir.
okay.
He's the co-host.
Yeah, Malik is the co-host.
I've gotten access. Thank you. But I'm still
Okay,
able
thank
to
you.
see the Q&A session at the bottom.
Maybe it's disabled.
Okay.
No, there is the chat board option, sir. Chat option. Question
Okay,
answer
okay.
cat it in a chat board.
Okay.
Okay.
So we have myself as the host, we have Kalro sir as the co-host, Jain sir, then Akshay sir, then Danish bhai, you are there, then Preeti madam is there, Saikat Malik is the co-host, Dr. Saikat Malik, and Krupa Ben is there, and Mahesh sir is there, Jankna madam. I think all those we require are here except our second speaker. I think he will join us later. Mamta Ben, where are you? Danish bhai, make Mamta Ben our co-host.
Ma'am, she's joining. She's in her cabin only. She's joining.
Okay, perfect. We request once again, all other participants to kindly keep their videos off during all the time, except the inaugural speakers and our lead main session speakers. Please keep your videos off at all the time so that we may not disturb the speakers, and kindly stay muted. Thank you.
Morning.
Alpana ma'am, Mamta madam in the meeting.
Yeah. Good morning, sir. Good morning, Carlos sir.
Good morning,
Good morning.
madam.
Good
Good
morning,
morning,
Dr. Akshay.
Dr. Mamta madam.
Good morning, Mamta.
Good morning.
Alpana ma'am,
Yes, sir. I think we should
can,
start now.
can we
We
start
are all
recording?
here.
Okay, ma'am. A very good
Alpana
morning.
ma'am, I am starting the recording.
Yes, sir. Please start. Please start. Yeah. Priti
Okay.
ma'am, start. Yeah.
Okay. A very good morning
Recording
to
is started.
A very good morning to one and all. On behalf of the LM College of Pharmacy and MG Science Institute, Ahmedabad, Gujarat, I, Dr. Preeti Verma, lecturer, LM College of Pharmacy, welcome you all to the 2 days international webinar on medicinal botanicals, present and future trends. Friends, we are already aware that many of the pharmaceuticals currently available to physicians have a long history of use as herbal remedies. Most of the modern prescription drugs contain at least one active ingredient derived from plant material, either obtained from plant extracts or synthesized to mimic natural plant compounds. Hence, the importance of botanicals in the medicines for therapeutic purpose is well established, and during the last several months of corona pandemic, the world has widely acknowledged them as effective and reliable drugs. Yet, the fast growing market of botanical medicines and other healthcare products throughout the world, policy makers, health professionals, researchers, and even the public are increasingly expressing concerns about the safety, quality, availability, and further development problems of these natural products. Realizing this scenario, we, the LM College of Pharmacy and MG Science Institute, jointly have tried to bring forth the best gems of researchers and experts on a common online platform in order to impart the knowledge to aspiring scientists about the major aspects of botanical medicines and their future prospects. Now, following the Indian tradition, let us begin the function with the virtual lamp lightning to express our deep gratitude to the Almighty. I request Rupa ma'am to share our video for the lamp lightning.
कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तुते दीपज्योतिर्नमोऽस्तुते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः.
Thank you, Rupa ma'am. Now, proceeding with the webinar, I shall now invite Dr. Mahesh Shabaria sir, Principal, LM College of Pharmacy and the convener of this webinar, to kindly greet the guests and audience and enlighten them with his golden words on this occasion. Over to you, Principal sir.
Thank you, Preeti ma'am. Respected Professor H. Kalu sir, Professor B.K. Jain sir, Mr. Akshay Charegaonkar ji, expert speaker of today's first session, Professor Mamta Shah, Professor Alpana Shukla, other faculty members and participants. I heartily welcome you all to this 2 days international e-webinar jointly organized by LM College of Pharmacy and MG Science College. LM College of Pharmacy is a premier pharmacy institute of India imparting quality pharmacy education at all levels, starting from DPharm to even PDF. To inculcate the culture of innovation and invention, the college has also established Atal Incubation Center, a state-of-art laboratories and workspace. And college is also a nodal center under Student Startup and Innovation Policy, which is given by the state government. This e-conference is organized on the theme of medicinal botanicals, present and future trends. The medicinal botanicals has already proven its importance, specifically in the present time of this pandemic corona, COVID-19. So this should not be the, as corona or COVID goes, this should not be the end for the medicinal botanicals. Now we have to take it to the next level, high, and we have to prove that these are the important constituents or the botanicals which are not going to or act specifically in the specific time only. It will go longer, specifically working in the therapeutics for the human ailments. The kind of speakers are invited in this e-conference. I am sure that all the participants will enhance their knowledge and gain from this 2 days e-conference. I compliment Dr. Mamta Shah and Dr. Alpana Shukla for working day and night to make this conference a successful one. Again, I welcome all of you to this international e-conference. Thank you.
Thank you, sir. Thank you, sir, for the enlightening words. Now, I would like to invite Dr. B.K. Jain, Principal, MG Science Institute, and the convener of this webinar to deliver a welcome address and throw some lights on the event with his kind words. Over to you, B.K. Jain, sir.
Thank you, Pratiben. Respected Professor Kalro, Principal Chabaria, our honorable speakers, Akshay Charegaonkar, Dr. Sheikh Bakhtiaruddin, Dr. Prem Kumara, and Dr. Shankar, conveners and coordinators of the webinar, and dear participants. On behalf of MG Science Institute, I welcome you all in this webinar jointly organized by MG Science Institute and LM Pharmacy College. MG Science Institute is one of the oldest institute in the Gujarat, started in 1946. The great personalities like Iron Man of the India, late Sardar Vallabhbhai Patel, first speaker of Lok Sabha, P.G. Mavlankar, great businessman, Kasturbai Lalbhai, and the first deputy vice president of India, Sarvapalli Radhakrishnan, played a very important role in the establishment of this institute. The father of Indian space science, Dr. Vikram Sarabhai, worked in this institute before the PRL, Physical Research Laboratory, was set up. The MG Science Institute was accredited at A level by NEC twice in 2007 and 2014. Institute is also ranked at 5-star level in the Gujarat State Institutional Ratings Framework, that is the GSIRF, for the academic year 2019-20. Many faculty members of the MG Science Institute, they are actively engaged in the research work, having fundings from the various funding agencies like Space Application Center, Department of Biotechnology, Department of Science and Technology, and more than 30 students are working for the PhD award under their supervision. The botany department of the institute is also engaged in the research activities on the antimicrobial activities of medicinal plants, where an approach is made to study the phytochemistry and biochemistry of the plants for their medicinal values. As you know, that World Health Organization said that more than 80% of the world population relies on the herbal medicine, or you can say traditional medicine, for their primary health care. And the medicinal use of the plants can be traced back over 5 millennia to the written documents of the early civilization of India, China, and Northeast. Among the estimated 2.5 to 5 lakh plant species, only a small percentage have been investigated phytochemically for their medicinal use. It means there is enough scope, there is a room for the further research. As you know, this in pandemic situation, the pharmaceutical companies are earning millions of rupees putting their herbal products in the market. Particularly, I name few like Giloy, that is the Tinospora cordifolia. Then we can say Ashwagandha, that is the Vidhana somnifera, and Tulsi ocimum. Of course, these are proved as immunity boosters. And I hope that within a few years, or you can say in coming days, nothing is wrong if the pharmaceuticals, or you can say the scientists, they will find out some herbal drugs against the COVID-19. Nowadays, the multiple drug resistance has developed due to indiscriminate use of commercial antibiotic drugs, commonly used in the treatment of infectious diseases. Sometimes these drugs are having many adverse effects on the host, including hypersensitivity, immune suppression, and allergic reaction. Therefore, there is a need to develop alternative antimicrobial drugs from medicinal plants. So once again, I tell you, a very small part of the plant species, or you can say the small percentage of the plant species have been explored for the medicinal value. Still, there is a room, there is a scope for the research, and we can find out, we can work a lot on this. I'm very much thankful to Dr. Alpana Shukla and Dr. Mamta Ben for combining pharmaceuticals and botanicals and jointly organizing this webinar. I'm also thankful to Principal Chabaria for this joint venture. And today's webinar will be definitely fruitful to those who love herbal medicines, and also to them, those who are actively engaged in the pharmaceutical research. I wish grand success of the seminar, and once again, I welcome all the participants. Thank you very much.
Thank you, sir, for the inspiring words. May I now invite Professor Dr. Mamta Shah, Professor and Head Pharmacognosy and Phytochemistry Department, LM College, and the convener of this webinar to give us an introductory note about the event. Over to you, Mamta ma'am.
I welcome you all. Good morning, Dr. Carlos, sir, Education Advisor, AAS, Dr. Chabaria, Principal, LM College of Pharmacy, Dr. B.K. Jain, sir, Principal, MG Science Institute, Alpna Madam, speakers of the day, Dr. Charigaonkar and Dr. Udin Sheikh, and my fellow faculty members as well as participants. I am given the responsibility of giving you some information about the proceedings of the conference, which is going to be there for 2 days. So looking to the scenario of today's situation regarding use of herbal remedies, we all see a huge upsurge in the utility of these medicines as a direct treatment or indirect treatment. But usually people see this as supportive medicine. But if we see actual chemistry part, then we can always say that botanicals are containing lots of potential biochemicals which are contributing towards their activity, and because of that, these phytochemicals which they contain, they are used as a lead drug source basically by the pharma industry world over. And huge number of laboratories as well as research institutes and industries are involved in the research on medicinal botanicals. And we hope that upcoming years would be very interesting to see more and more of surge and utility and fruitful utility of this kind of medicines. So I would like to say that keeping this view in mind, we, myself and Alpina Madam, planned this conference, e-conference, to involve those who are working in the field of botanicals, to give them the knowledge, not only botanicals, but I would say phytopharmacology, phytopharmacognosy, etc. So those scientists who are working in this field will be getting benefit about how to go for the actual research, how to strategically go for the research and get kind of interesting results which would be useful to the society. And that is how we planned this. And the theme actually came spontaneously from both of us, that we should be going for something on botanicals. So I said we can always go for medicinal botanicals and their present and future trends. So we know what is the trend and what is the when future is going to be for this medicinal botanicals. I would say that keeping these things in view, I invited speakers from all over the world. Myself and Alna Madam did some planning and what to do and how to invite and whom to invite. And we both planned speakers from Sri Lanka, Bangladesh, UK, and even India, the Indian speaker who is going to talk on HPTLC. So the 2 days are going to be very interesting because various facets of research aspects of medicinal botanicals are going to be taken up by the speakers, and it will be really interesting and learning sessions throughout these 2 days. So I hope that you will all be learning and enjoying the sessions of the conference we have planned. Thank you so much.
Thank you, Mamta ma'am, for giving a nice introduction of the event. Well, we deem it a great honor to have with us Dr. A.H. Kalro as today's chief guest. It gives me immense pleasure to introduce Sir. Professor Dr. Kalro is presently an academic advisor of Ahmedabad Education Society. Sir is an educationist with more than 50 years of experience in academic institutions in India and abroad. He has contributed extensively to educational policy and establishment of educational institutions. He was a member of the Second Administrative Reforms Commission of the Government of India. He was former director of IIM Kozhikode and has also served as chairman at Ravi J. Mathai Center for Educational Innovation at IIMA. Sir has been an assistant professor at the University of Minnesota also. Professor Kalro obtained his BTech Honors degree in Electrical Engineering from the University of Minnesota, USA. Sir is the recipient of several scholastic and teaching excellence awards. One of them is the Lifetime Achievement Award from the Indian Society of Operations Management in diverse areas, including management of educational institutions in reputed international and Indian journals. He has also co-authored 3 books and has extensive consulting experience with public and private sector organizations, along with state and central governments and international agencies like Ford Foundation. He has traveled extensively for more than 30 countries and has taught in programs in Japan, Kenya, and Sri Lanka. We are very happy to have Dr. Kalro with us today as our chief guest. So I welcome Dr. Kalro and I request Sir to say a few words on the occasion. Over to you, Kalro, sir.
Thank you very much, Preeti, for the generous introduction. And a very good morning to everybody present today at this webinar, international webinar on medicinal botanicals, present and future, being organized by the LM College of Pharmacy and the MG Science Institute. Both these colleges are renowned for the contribution that they have made to higher education in their respective fields. And in fact, they were established just 1 year after each other. MG Science in 1946 and LM College of Pharmacy in 1947. The Ahmedabad Education Society, of course, was established much earlier in 1935 with inspiration from late Sardar Vallabhbhai Patel, who was very keen to ensure access to education of all people, children who were in this region. His idea and the vision of late Kasturbai Lalbai led to a strong foundation being laid for the Ahmedabad Education Society in 1935. Its first educational institution was set up in 1936. And of course, today, it is the promoter of 2 universities, 5 colleges, 3 schools, and another teacher training college. In fact, the reputation, I would dare say, of the Ahmedabad Education Society has been built up assiduously over the years by its institutions and the prestige that they commanded and the choice that they provided to both students and parents in this region to further pursue their academic endeavors. Now, I'm particularly pleased today that we have 2 of these well-known institutions collaborating with each other to organize this webinar, pharmaceuticals and botanicals. Now, this is something that we have been trying to do for a long time. And gradually, we are beginning to enjoy the fruits of the efforts that we have put in for colleges to collaborate with each other. Because, 1 + 1 is not 2, 1 + 1 is 11. And that is what we're now trying to demonstrate to the world at large, that together we can achieve much more than what we can do individually. Indeed, the corona pandemic has also made things a lot easier for us in a way by helping us to leverage technology to be able to work together and also bring at a common platform resources and eminent persons from around the globe to share their knowledge and wisdom with us. And today is an example of that, because we have speakers drawn from different countries. We have participants who are registered from different countries. And of course, we will be spreading knowledge to various corners of not only this country, but other parts of this world. Now, plant medicine is not new, or botanicals for use in medical purposes is not new. It probably dates back to the history of mankind, when plants were used as the only remedy available to address a large number of problems and injuries that the body faces. Indeed, the Rig Veda itself has reference to a large number, a very large number of plant medicines and herbal medicines. And not only that, I think all of us are familiar with the mythological story, you may not call it mythological if you like, of Ramayana, and how Lord Hanuman goes all the way to the Himalayas to fetch the Sanjeevani booty to be able to save the life of Lakshman. He had just 24 hours maximum to live. And in that 24-hour period, Lord Hanuman went all the way from Lanka to the Himalayas. And since he could not identify the booty, he brought the whole mountain to Sri Lanka so that Lakshman could be saved. Indeed, we have today, as was mentioned by Dr. Jain, close to, I think, at least 5 lakh plants which have potential value of medical use and other purposes related to medical use. But as of now, barely 5,000 have actually been documented, tested, and submitted to some kind of standardized processes which will enable us to understand the outputs of products that we derive from them, which can be used for specific purposes in amounts and dosages which can be calibrated. We have a long way to go, both in terms of adding to the pool of plants and herbs which we can basically draw on, and secondly, to develop them further to actually put them to use. All our speakers have made a reference today to what the corona pandemic has done to drive home the importance of these products. But you know, I think what has begun with the corona pandemic will actually end up in scaling heights which we could never have imagined. Today, for example, the total exports of plant medicines and herbal products is just about $500 million. Very small indeed compared to the size of the pharmaceutical industry that we have worldwide. And even this figure is very small compared to the size of some of our pharmaceutical companies in India, which are into a big way in exports. And as you all know, the export of pharmaceutical and medicinal products from India is probably amongst the highest in the world. And this, by the way, includes patented products which are manufactured under license or under our own patents. We have a long way to go. And I'm sure that with webinars and international conferences like these, we are making a good beginning, and the institutions which are collaborating will basically continue to make this endeavor shine brighter and better for the future of the citizens of the world. I would be feeling in my duty if I did not thank at this point of time our esteemed speakers of the conference during the 2 days. I think Dr. Akshay Chargaonkar is actually present. My namaskar to him. I did not see any of the other speakers, but a warm welcome to all of them. A very warm welcome to all the participants of this conference also. And I have every feeling that we're going to take home a lot of knowledge with us, which we will put to use in not only pharmacognosy with Dr. Mamta Shah heading this in LMCP, but also in botanicals with Dr. Alpana Shukla heading this in the MG Science Institute. I must draw your attention to the fact we have two eminent ladies leading the effort. The men should take note. So thank you very much, everybody. Of course, not to mention Dr. Preeti Verma, who's also been playing the host and also belongs to, I think, the field of cognosy. Am I right, Preeti?
Yes, sir.
Yes, okay, good. So thank you very much, all of you, for the efforts that you have put in, for what you're doing for the Ahmedabad Education Society, your institutions, and indeed creating a position for yourself in the global population of educational institutions, which will do everybody proud. Remember, we now have to address all the challenges of NEP 2020 also. So this is the first foundational step that we are taking. We do many, many more things, and I look forward to contributions of all our faculty from the different colleges in this effort. But today, a special thanks to Dr. Jain, Dr. Chabria, Dr. Mamta Shah, Dr. Alpana Shukla, Dr. Preeti Verma, our resource persons, and all the participants of this webinar for giving a boost to our effort.
Sir.
You are the engine, they are the fuel. So thank you very much and all the very best to all of you.
Thank you, sir.
Thank you, sir.
Thank you, sir, for the encouraging words and for sharing your views about the upsurge of herbal medicines. You rightly correlated the herbal medicines with their origin in India from ancient times. Thank you very much, sir. And now we are heading towards the end of the inaugural function, but before that, it is the time to recognize the efforts behind the organization of this webinar. So I would now request Professor Dr. Alpana Shukla, Professor and Head Botany Department, MG Science Institute, and the convener of this webinar to deliver the vote of thanks. Over to you, ma'am.
Thank you, Dr. Preeti. It is my privilege to propose the vote of thanks, the formal vote of thanks. First and foremost, I thank both our principal sirs, Dr. Mahesh Chabaria sir from LM College of Pharmacy, and my own principal, Dr. BK Jain sir from MG Science Institute, for constantly supporting us in all our activities for the students, and always being there for us no matter what we are planning. And this goes for both our principals. We thank you from our bottom of the heart. Thank you, sir. We would like to express our deep gratitude to Professor Kalro sir, our academic advisor, who always brings the best in us, brings out the best in us. He's always there to make time for us and being with us at every occasion whenever we invite you, sir, and giving us your valuable counsel. Thank you very much, Professor Kalro, sir, and we thank our management also. At this point, I would like to thank, on behalf of all of us conveners and organizers, our resource persons for today and tomorrow. Without them, this webinar would not have been possible. They willingly accepted our invitation and they are with us today and tomorrow. So let us learn the maximum that we can and imbibe their knowledge, share their knowledge. Thank you, all the stalwarts of this particular science. Thank you very much to our resource persons. We would like to thank Institute of Engineering and Technology, Ahmedabad University, for providing us this platform and providing all the IT help that we need. We would like to thank also our coordinators, Dr. Preeti, Dr. Jankna, and Dr. Krupa. And we would like to thank our office staff who have always been with us in all our activities and endeavors. At this point, I would like to thank all the participants who have made time today, this morning, and who are going to join us tomorrow also. Thank you very much for being with us. Last but least, not the least, I would like to thank all those people who have helped us directly or indirectly in any which way. Thank you very much.
Thank you, ma'am, for the beautiful vote of thanks. Now, without further delay, we'll proceed towards the scientific sessions of day one. Our first speaker is Dr. Akshay Sharegaonkar, who is going to deliver a talk on modern HPLC instrumentation, automation, and pharma applications. For that, I would like to request Dr. Jankina Rathore, Assistant Professor, Botany Department, MG Science Institute, and one of the coordinators of this webinar, to introduce Dr. Sharegaonkar and take over the further proceedings of today's session. Over to you, Jankina ma'am.
Thank you, Preeti ma'am. I consider it is a great pleasure to introduce you all to our first speaker, Mr. Akshay Sharegaonkar, sir. Sir did his BSc in Chemistry from Rochester Institute of Technology in New York State in 2008. He was actively involved in non-academic activities as well. Subsequently, he worked in Beaker Corporation near Boston for a year, working on polymers in semiconductor housing contest. He resigned in order to return to India to pursue higher studies as well as to manage his family business. He joined Ancrom in October 2009. At Ancrom, he is Director of Business Development for all aspects of business, including the India specific HPLC Application Research Laboratory. As a part of his responsibilities, he has been visiting Kame, Switzerland, and Mitsubishi Electron, Japan. He has delivered numerous lectures on HPLC all over India. He was a co-author for a poster on HPTLC MS put on at ASMS 2015 at South Louis, USA. Sir, it will be a great pleasure for all of us to listen to you today. I request, sir, to please start your presentation. Over to you, sir.
Thank you very much for that very warm and generous introduction, madam. Let me begin by sharing my screen. So good morning, everybody. I hope that my screen is visible to all.
Yes, sir.
Okay, great. Thank you very much. Thank you very much again for having me and on this wonderful webinar with really very timely and important topic, that is botanicals, the present and the future, because this COVID pandemic has shown us that no matter how much the pharmacy sector has gone ahead, we are still helpless against this COVID. So perhaps plants have the answer. So thank you very much for to all the dignitaries and to all the participants. So let me begin my presentation by describing how we'll break this presentation into three parts. First, we'll talk about HPTLC, the technique, the regulatory aspects of the same, the hyphenation, which is the latest techniques, which are HPTLC combined with other techniques such as HPTLC MS, HPTLC bioautography, because after all, the topic of this webinar is HPTLC present and future. So regarding the hyphenation, that is the future of HPTLC. We will also talk about the automation, and we will spend most of our time talking about the applications of HPTLC. Now, my colleague, Dr. Saikat Malik, he is in the chat box. If during my presentation, any of you have any questions on the presentation content, please feel free to type in the chat box, and Dr. Saikat and his team sitting in our laboratory will be very happy to reply. So I'd like to begin by telling you that we have specially designed this content for the Zoom webinar format. We know many of the students especially are logging in from your mobile phones. So we try to keep the text and the images as clear as possible that even a small screen can properly display them. Now, as Dr. Kalro sir said, that 1 + 1 does not have to be 2, it can be 11. I'd just like to tell you that all of the HPTLC content that you're going to see now is available in our laboratory. We offer free training to any of the students or teachers, postgraduate students and teachers, professors who would like to get trained at our laboratory in Mumbai. Our laboratory is now open with the proper corona safety procedures. Training can be arranged at mutual convenience. You can write an email to us at hptlc@ancrom.in, or you can call or WhatsApp on this number that you see on your screen. We'll be very happy to give you training and certification. We have just now opened a brand new HPTLC application center just last year, which is a very fortunate time to do that because we were able to finish the project before the lockdown. And we are all very much welcome to take advantage of it. We have capacity for up to 20 trainees at once. So the reason I'm telling you this at the start of the program is that if you feel any of the applications that I'm going to show to you are of interest to your research work, please feel free to get in touch with us. We also have a YouTube channel where we have put in educational videos, training videos. You can just search for Ancrom on YouTube and our channel will pop up. You are free to use that content in your examinations or your syllabus and so on. So that content is available free of cost to anybody who wants to use it. Now let us begin the technical aspect of the program. HPTLC stands for high performance thin layer chromatography. Now the main word here is chromatography. You know, all of you know that the various types of chromatography are HPLC, GC, HPTLC. All of them have one aim. All chromatography is the science of separation of mixtures. Now, a mixture could be anything. It could be a herbal drug. It could be an extract of a plant, not made into a drug yet. It could be a pharmaceutical pill. it could be a food, it could be something, a packet of suspicious white powder caught at the airport. It could be anything. The science of separation of mixtures means that once we separate the mixture, we want to identify what is in my mixture. What is the particular useful API in this particular formulation? You want to quantify, you want to know what is the quantity of the, for example, the adulterant in my material or the impurity. Is it 1%? Is it 0.1%? And the third thing is that we want to identify, after identification, sometimes we want to separate a appreciable quantity of uh, that particular component of the mixture. For example, if your plant extract has a useful secondary metabolite, which you want to do further studies on, then you want to isolate 10 or 15 milligrams of that and send it for NMR or send it for further testing for microbial activity and so on. So that is microfiltrative analysis. Within identification also there are two types. One is comparative identification, where we have a standard and a sample. We compare the signal that we get from the standard to the sample. If the signals are the same, we say, yes, this product has been identified. It is the same thing as the standard. But there is also absolute identification where you do not need a standard. Some techniques either have rules for interpretation of the data or they have a built-in library, which is the case in terms of TLC-MS, or in terms of NMR, where there is a rule for interpretation. And without a standard also, you can identify. So these are common for all types of chromatography. It is the science of separation of mixtures. Now let's focus on our topic at hand, TLC or HPTLC. Now I'm showing you TLC analysis of Asian ginseng. Now I'm speaking directly to the students in the audience right now. This is what the TLC you do looks like, isn't it? You take a strip of TLC plate, you make a line with a pencil where you are going to apply your samples. You apply your sample as a round spot with a capillary containing the sample. Then you put the TLC plate in a beaker containing the mobile phase, and the mobile phase travels up the length of the plate by capillary action. When the mobile phase reaches the particular development distance, you take the plate out of the beaker and either you look at it under UV light, or what is happening in this case, you put the plate in a tank containing iodine, which is a derivatizing agent. Iodine has the property of sublimation. It goes directly from solid state to gas phase and it sticks onto any organic compounds. And you can see this is what you get, right? I'm not in front of a live audience, but this is what a regular TLC used to look like. Now, you can see several problems. एक तो ये जो standard है, is it possible to determine if that standard is the same as this? No, because the RF value does not properly match. Secondly, due to the iodination, I can only see one color. Everything is purple. And third thing is that there is no separation of the particular secondary metabolites here. Now, let us look at the same sample, Asian ginseng, but under HPTLC, high performance TLC, not regular benchtop TLC, and see what the difference is. Now you see the difference. I can separate a very, very large number of secondary metabolites. You can see here that there is baseline separation between all of these peaks. Secondly, I have chosen a particular derivatizing reagent which causes different colors to appear based on what is the type of phytochemical present in that particular zone. And third thing is that you can see I have 1, 2, 3, 4, and 5 standards, and they are all well separated. And I can very easily see which one of those standards or the markers are present in my sample. So you see the huge difference between TLC, which we used to do, that is the old way, and HPTLC, which is standardized, and high quality analytical results are obtained by it. Now, many of you are students of pharmacy colleges. You know that there are many, many monographs in the Indian pharmacopoeia, in the European pharmacopoeia, US pharmacopoeia, where they say use a 20 centimeter by 20 centimeter bada wala HPTLC plate. But that is all TLC. When we have those methods in HPTLC format, what we do is that we shrink the entire method down. Instead of 20 by 20 centimeter plates, HPTLC is always done on 20 centimeter by 10 centimeter plates. So the same sample or the same time, it used to take about 90 minutes for a development of 16 to 17 centimeters. Now that same resolution can be obtained in 30 to 35 minutes by developing up to only 7 centimeters. So we have miniaturized the entire process from TLC to HPTLC. Now this is the table of differences between TLC and HPTLC. I am not going to go through the whole table. This presentation will be available to you if you wish to go through it. I want to focus on three things. One is the primary focus of TLC used to be on simplicity and low cost, जिसको हम लोग हिंदी में फटाफट बोलते हैं. फटाफट बेंच टॉप पे मुझे रिजल्ट चाहिए. For that, I will use a capillary. I will quickly take a छोटा सा पट्टी of TLC plate. I will touch the plate with the capillary. I will put in a beaker. I will see under UV cabinet. So फटाफट simple and low cost method I want, that is TLC. Whereas the focus of HPTLC is on reproducibility and separation power. You want to have good separation and you want the results that you get to be reproducible. उसमें कोई variation नहीं होना चाहिए. आप same experiment 10 बार करोगे तो you should see the same result 10 times if the sample is the same. The target of TLC was rapid preliminary results. Again I said, फटाफट मुझे bench top पे, if I'm doing a reaction, I want to see quickly if my reaction is A plus B makes the product C. So I can see the number of spots on the TLC plate very quickly and I can see if A plus B equal to C reaction हो रहा है या कुछ side chain बन रहा है या reaction is not going to completion. I can quickly do it on a bench top. That was the focus of TLC. But in HPTLC, the focus is on reliable analytical results. What we want to do is कि ऐसे first class result मिलने चाहिए, which will give, you can submit to any regulatory authority across the globe. They will accept it. You can submit it to any prestigious journal. They will accept it. So the target of HPTLC is on reliable analytical results which are reproducible. Whereas TLC where the focus was ki fatafat karo, or if you want more data, go to the other technique like HPLC. But HPTLC is meant to be an end in itself. And the instrumentation for TLC also used to be almost no instrumentation. एक UV cabinet है, एक iodine chamber है, beaker, capillary. But HPTLC can be done using simple instrument also. It does not require any fancy instrument. If you want fancy instruments, they are available, but not necessary, which we will talk about in the next few slides when we talk about the regulatory aspects of HPTLC. The practice of HPTLC is very simple. First, you prepare your sample. Sample preparation is also simple because you don't have to worry about the solvent that you use. The HPTLC plate is disposable. So you can use a very highly acidic solvent to dissolve your sample or very basic or whatever you need. Then we apply the sample to the plate in a controlled, uniform and reproducible manner. In HPLC or GC, this step is called injection. Because in HPLC or GC, there is a column into which the sample is physically injected because the column is closed. And in HPTLC, the plate is open. उसमें कुछ inject नहीं हो रहा है. We are just spraying the sample on the plate. So we call that sample application and not sample injection. Then we do the chromatogram development. That is to say the plate is put in a tank containing the mobile phase and the separation happens. Then we visualize the plate. That is, we look at the plate under 254 nanometer light, 366 nanometer light, white light. We take photographs of the plate. Then we do densitometric scanning, which is the main information gathering step. So which we will see in the next few slides how this is done. Now, we talked about the differences between TLC and HPTLC. So I won't go too much into this slide. Now let's talk about HPTLC and why it is a preferred technique for herbal laboratories. पहली बात तो ये है, herbal samples have a lot of matrix because the samples are, extracted from plants. So you have chlorophyll, you have cellulose, you have fatty material, you have a lot of matrix. It is not a very clean sample coming from a factory. It is coming, basically you are extracting the juice from plants. So there is no guarantee that the sample will be extremely clean. But in HPTLC, what is happening is that you do your analysis on a fresh HPTLC plate every time. Whereas in HPLC, you have to use the same column, 50 times, 100 times, 200 times. और वो कॉलम खराब हो जाएगा तो इट इज अ ह्यूज कॉस्ट. सो यू हैव टू वरी कि इज माय chromatography instrument going to be contaminated, is my column going to be contaminated. HPTLC में ऐसी कोई बात है नहीं. एनालिसिस के बाद आप प्लेट बाहर रखने वाले हो, नेक्स्ट एनालिसिस के लिए यू आर गोइंग टू टेक अ फ्रेश प्लेट. Then it is a very simple and fastest technique because HPTLC में you have one applicator, one for development, something for photo documentation, something for derivatization, something for documentation. So you have multiple instruments and they can be used separately at the same time. Whereas in HPLC or GC, you inject the sample from one side and nobody can use that machine until the results come out from the other side. HPTLC में ऐसे नहीं है. Many people can work on the same system in parallel. In our lab also, we have 14 analysts, but we have only 2 applicators, 1 scanner, many development times. तो भी हमारे लैब में आपको ऐसे नहीं दिखेगा कि मैं रुका हूँ यार, अभी मेरा टाइम कभी आएगा, ये मशीन बिजी है. Never. You can plan your work, so it becomes a very, very fast and simple technique. There is very low running cost and maintenance as well in HPTLC because there is no high temperature and no high pressure used anywhere in the system. Everything is done at normal room temperature and pressure. So therefore, you don't have to worry about oh pump ka seal kharab ho gaya, pump kharab ho gaya, the leakage hai, back pressure hai. None of those things affect HPTLC. And due to that, you have a low maintenance and you can do up to 15 samples on the same plate using hardly 15 to 20 ml of your mobile phase solvent. वो भी the solvent can be very, very cheap AR or GR grade solvent. You don't need expensive grade high purity solvent like HPLC or MS grade. तो solvent भी कम लगेगा, उसका level भी lower level का solvent use कर सकते हैं, so it comes a very low running cost. Per sample, you are using hardly 2 to 3 ml of mobile phase if you do 15 samples on the plate. Then there is double confirmation of results. That is to say the plate itself is your result. HPTLC is a very visual technique. I am going to show you so many photographs of plates. Oh, photograph देख के आधा काम हो जाता है. Of course, then you get the printed data from the instrumentation, but there is double confirmation. So let's look at HPTLC. I know that there is a large academic crowd, so I put this plate in here specially. HPTLC features modular hardware. I told you that HPTLC में ऐसा होता है कि you can one person is doing application, other person is doing development, third person is doing scanning. Multiple analysts can work on the same system, so it is very applicable for an academic lab. जहाँ पे मैं privately एक HPLC लेके बैठ नहीं सकता हूँ. I have to share it with all my colleagues and other students. Then the software that I showed you or I'm going to show you is database oriented. Everything is stored in a SQL database. तो मैं मेरा यूजर नेम और पासवर्ड डालूं तो मुझे सिर्फ मेरे फाइल दिखेंगे. I cannot see other students' work, but the professor who is at a higher level, they can see everybody's work. So each analyst can keep track of their files. And you know, when you are working in research, MSc, PhD, it's not a 1 week job. It takes 2 years, it takes 3 years, 4 years. We have many files. 4 साल का मेरा analysis. It is very easy to keep track of these things because it is in a database. You can just search. If I'm searching for, let's say I'm working on haldi, kurkuma longa, I just type in kurkuma. और मैंने 5 साल में जो kurkuma का काम किया वो सब मुझे दिखेगा. And there is solid Enchrome support since 1978. Now ऐसा होता है in the academic environment especially that students, you know, they get their degrees, they go away. Then a fresh batch of students comes in. So there is sometimes a lack of continuity. तो कभी-कभी हर साल ट्रेनिंग की जरूरत पड़ती है या 2 साल के बाद. या if there is a new PhD student, they can pick up the phone and call us and say, I have just joined. ये instrument मुझे दिख रहा है. I want to use it. How do I use it? So then we will be able to provide you with all the training you need. तो machine सिर्फ supply करके it is not enough. You have to keep it running for 10 years, 20 years, 25 years. And to keep it running, you need service support and you need training support, which Enchrome is able to provide. So that's why I put this slide especially because I know many of the people are from research labs. Now, let us move our topic to the regulatory acceptance of HPTLC. This is quite a new topic because ये 4-5 साल से ही हो रहा है. That HPTLC has become an official part of the US pharmacopoeia, the European pharmacopoeia, Indian pharmacopoeia, the WHO, World Health Organization guidelines for herbal medicines, all of these have accepted HPTLC as a completely regulated technique. USP has two chapters called Chapter 203, which is a very, very simple 2-page chapter, and Chapter 1064, which is more, it is 11 pages. Chapter 203 describes all the parameters you have to standardize if you want to call your work as HPTLC. We'll see that in the next slides. The European pharmacopoeia has basically harmonized with the US pharmacopoeia. They have basically said कि US का जो है वो हमको acceptable है. The Indian pharmacopoeia is not completely harmonized, but they are moving in that direction. So let us take a look at what is the USP general chapter. This is very important if you want to get into manufacturing, if you want to get into exports of herbal drugs, you will have to comply with this chapter. The chapter is called HPTLC for the identification of materials of botanical origin. Its purpose is to provide a standardized HPTLC methodology to distinguish official botanical materials from non-official ones. मतलब adulterants हो, या substandard हो, या other subspecies which are not accepted. You can distinguish the main material from all of these other potential adulterants. It specifies the instrumental requirements for most steps, but 2 page का chapter is सिर्फ. And we'll see what steps are specified. So they have specified the plate layout. उसमें लिखा है कि प्लेट को अगर आपको सैंपल को अप्लाई करना है, how much distance to leave from the left side before you apply your first sample? What should be the length of each band that you apply your sample? What should be the gap that you leave between 2 samples on the plate? How much development distance should be there? How much should be the height of the mobile phase in the developing tank? Everything is specified by them. In a simple chapter, they have said you have to apply rectangular bands and not spots. जो हम लोग spot लगाते हैं with a capillary, that is not allowed by USP. The most important thing they have told is that you should do all of this work at relative humidity of 33% in the tank. Now, this is because HPTLC is an open technique. So when there is a change of humidity in the air, there can be a difference in the amount of moisture that has adsorbed on the surface of the HPTLC plate. And that can lead to a change in the chromatographic activity, a change in the number of theoretical plates, which you may have learned about in your coursework. So, ये सब eliminate करने के लिए, you know, in India, in Gujarat, in summer, it is very dry. In Mumbai, it is extremely humid. In other parts of the country, the situation is different. So, to keep the chromatographic quality the same year round, and no matter where in the country or in the world you do your work, 33% RH पे काम करना है. There is an instrument to achieve 33% humidity in your developing tank by CAMAG, but it is easy to do it without an instrument also. You take a desiccator, you fill the bottom of the desiccator with a saturated solution of magnesium chloride. You close the desiccator for about 1 hour, and then you put your tank or your beaker and you do the development in that desiccator. तो भी आपको 33% मिल जाएगा. So the USP has made it simple to comply. You don't have to buy any fancy equipment. It will make your life easier, but it is not a necessity. So these are the USP HPTLC parameters. The plate layout is specified, developing distance is specified, relative humidity is specified. And now let us look at what a USP monograph looks like. This is a USP compliant monograph of a Chinese herbal medicine component called Ganoderma mushroom. It is known as reishi mushroom. It is very famous in traditional Chinese medicine. So I want to draw your attention to this area of the plate. So this is the HPTLC monograph of that particular Ganoderma mushroom. So you can see there are 19 tracks in this monograph. Track number 1 that you can see here is these are all the markers or the secondary metabolites present in the Ganoderma mushroom. So there is ganoderic acid, ganoderianic acid, ergosterol, and so on. Track number 2 to 7, which are marked with yellow brackets, these are all the acceptable material of Ganoderma mushroom. So you can see already that they have put not one example of fingerprint, they have put 6 examples. This is because any herbal medicine, be it a plant or it can be any other thing like a mushroom also, these are all living things. They have their own life cycle. So they have a chemical variation in them. If you harvest them from different areas or if you harvest them during different seasons. So your monograph has to cover the entire life cycle of this plant or mushroom in this case. So we show not one example, but 6 examples. ये कोई भी fingerprint इसमें से acceptable है. Because these are all botanical or authenticated reference materials of Ganoderma mushroom. Now, ये हो गया acceptable material. Now you can see track number 8 to 19 show you the unacceptable material. So they show you the adulterants. So Ganoderma or lucidum fruiting body is the official Chinese herbal medicine mushroom. So then there are other subspecies of Ganoderma like Ganoderma sinensis, Ganoderma tussace, Ganoderma ka or B subspecies. Those are all unacceptable. So this monograph shows you 6 examples of good Ganoderma mushroom and it shows you 11 examples of bad Ganoderma mushroom. So you see, that is what the HPTLC concept is. You should be able to do quality control by saying what is a good material, what are the markers in it, and you should be able to distinguish it from adulterants. Because once you dry and powder this mushroom, everything becomes a brown powder. So the HPTLC fingerprint is the only way to really tell the difference between this brown powder and that brown powder. So this is the example of a HPTLC monograph using standardized regulatory HPTLC. What 3 things I want you to focus on is, one is it shows all the markers of that particular plant or mushroom in this case. It shows you multiple examples of good acceptable material and it shows you multiple examples of bad unacceptable material. So that is a HPTLC concept. HPTLC regulatory bodies ask for this kind of monograph. Now let me show you an example of work done over a period of 1 year in Chemag's laboratory in Switzerland of curcuma. So you can see from here to here, all of this is various samples of curcuma, and you can see that they are all looking very, very, very similar. This shows you the reproducibility of the technique. मेरा आज मूड खराब है तो मेरा chromatography खराब है, मेरा मूड अच्छा है तो chromatography अच्छा है. These things do not happen in regulatory HPTLC. Everything is standardized. There is no room for error. So you can see that all the work that is done over time looks exactly the same. Now you can imagine this plate was done in February. This plate was done in, let's say, August. This plate was done in October. But they all look exactly the same, showing you the reproducibility of regulatory modern HPTLC. Now, one more thing is that I have not made this graph or this multiple image in Photoshop. I told you already, HPTLC has a database. So मैंने curcuma search किया तो all of the work that I have done in curcuma, it shows up like this, and this is an output directly from our software. So you can compare your own work across time. You can compare your work to other people's work, and it gives you a very beautiful comparison view inside the software. Now let us look at how we use HPTLC to distinguish between subspecies of the same plant. Now curcuma longa is our haldi. And there is another curcuma species called curcuma aromatica, also known as curcuma xanthorrhiza, which is present in Southeast Asia, which we don't use as haldi. So I want to draw your attention to the leftmost column, this one, the leftmost plate. This is 100% pure haldi. Now you can see that 100% pure haldi has these 3 curcuminoids: curcumin, methoxycurcumin, and desmethoxycurcumin. And on the right side, this is 100% pure adulterant material. यहाँ पे हल्दी नहीं, it's all adulterant. So you can see that this third curcuminoid is completely missing in the adulterant material, which is present here in the actual haldi. And this purple zone is present only in the adulterant material, and it is not present in haldi at all. So now let's take a mixture of 50% pure haldi and 50% aromatica. So उसमें क्या होता है? You see this purple zone has shown up, but less bright because it is only half of the material. And this curcuminoid are also shown up because that is from the pure haldi. Now let's take an example of 90% pure haldi with 10% adulterant. That is here, 10% aromatica, 90% haldi. So you can see this curcuminoid band is very strong now because that is 90% pure haldi. And you can see very faintly this purple band has shown up. That is from the aromatica. So we can not only detect adulteration, we can detect the quantity of adulteration, because you can see as we go on increasing the concentration of the adulterant, the purple band gets more and more prominent. And at the same time, this curcuminoid band gets less and less prominent. So we can detect adulteration and we can detect. Now, the thing is that I would like to tell the organizers to please tell me how much time I have left, because I'm a little confused about how much time I have, so that I can decide how much deep to go into the instrumentation. Dr. Montaman, can you help me out? How much time do I have left? Okay, okay, let me go on. And if there is any, you can let me know.
Yeah.
Oh.
Just a minute, Dr. Akshay. We are running over the time, so try to cover it up like within 5 minutes.
Okay, fine. Okay. So now.
5 minutes, okay?
Only about 10-12 minutes. Will that be okay?
Okay.
Okay, fine. So now, since I have 10-12 minutes left, I have a section of HPTLC instrumentation in this presentation, but I will skip past that because LNCP has a very beautiful new HPTLC system. Our lab has a nice HPTLC system, so you can come and see it in our lab. I would like to spend more time focusing on the applications thereof. So I would just like to show you what a complete HPTLC system looks like. You have an automatic applicator to apply the sample to the plate. Then you have an automatic development chamber to develop the plate with control of humidity. Then we have automatic derivatization. We have a scanner which gives you peaks the way you would get from HPLC or GC, where the area under the peak or the height of the peak corresponds to the concentration of that particular component in your mixture. We have a photo documentation system which takes high resolution photographs of the plate under white light, 254 and 366 nanometer light. And we have the hyphenated technique called TLC-MS, which gives you the mass spectrum of any of the zones of interest from your plate directly from the TLC plate without going into LC. This is a complete system for HPTLC. Now, I want to speak about the importance of HPTLC, especially in the field of botanicals, where there are about 10,000 HPTLC systems all over the world, out of which about 1,000 are in India. More than half of those are for herbal analysis, because HPTLC is the gold standard when it comes to herbal analysis. And there are several hundred government labs and pharma companies and so on doing herbal work and herbal manufacturing on HPTLC. And HPTLC is a major part of any lab which is doing herbal analysis. Now, as I told you, since we are running a bit short on time, I would like to skip past the instrumentation aspect and move directly to the hyphenation side. Instrumentation तो हम लोग बात कर ही लेंगे. Okay, so now let us talk about the hyphenated techniques, which is TLC-MS is the first hyphenated technique. The TLC-MS, as you can see, this instrument has two capillaries. One capillary is connected to the LC and the other capillary is connected directly to the mass spectrometer. Then there is a head of the instrument which is positioned manually and comes down onto the plate on the zone of your interest. And then the solvent goes into the incoming capillary. It dissolves whatever material of interest is there, and it goes out directly from the plate into the mass spectrometer. This machine is simple. It does not have a MS built in. It requires you to already have an LC-MS in your lab, which it connects to. And there is a 2-micron filter at the outgoing end, so that anything that any silica gel or any other material which was on the plate will not be going into your MS. So you can see that using HPTLC, you can get a good separation, and you can also see how you can now work with unknowns also by doing mass spectrometry. Now, for example, this is an example of tadalafil in Ayurvedic Rasayan. Now, tadalafil is a analog of Viagra, which is sildenafil. Many times, herbal medicines are contaminated with allopathic APIs. Herbal Viagra में real Viagra, herbal painkiller में synthetic painkiller like non-steroidal or steroidal anti-inflammatories, and so on. So this is an example of HPTLC analysis followed by MS for catching this adulteration. This is a plate of that rasayan which we were able to separate, and we were able to get the positive identification of tadalafil by using TLC MS. Now we showed a joint poster with Shimadzu. We have a tie-up with Shimadzu because we don't have an MS in our lab. We take our TLC MS to Shimadzu's lab. So we published this work, characterization and isolation of quinones from teak leaves, Tectona grandis. So Ancrom took care of the HPTLC work of the separation of these quinones, and Shimadzu took care of the MS work. So this system is in the market. We have supplied about 40 to 50 of these all over India, and they are doing very well for, especially for research in unknowns such as new product development and so on. Now, the second type of hyphenation I would like to talk to you is the bioactivity detector, which uses a bioluminescent bacterium called Vibrio fischeri to detect any toxicity in your sample. So let's see how it works. What it does is that it uses a bioluminescent bacterium called Vibrio fischeri, which has three characteristics which make it useful. Ek to, this bacterium is bioluminescent. When there is a healthy colony of these bacteria, they emit a light. Secondly, these bacteria are very easy to kill. आपके सैंपल में कुछ भी टॉक्सिक कंपोनेंट होगा तो वो बैक्टीरिया मर जाते हैं उसकी वजह से. It could be organic toxicity or inorganic toxicity. The third thing is that this bacteria is completely safe to handle. You don't need any special microbiological facilities to handle it. It is completely non-toxic or harmless to humans. तो हम लोग क्या करते हैं? We make a HPTLC plate. We develop it as usual. Then we dip that entire plate in a tank containing this bacteria. Then we incubate the plate for 2 hours. तो जहां पे plate पे bacteria है, वहां से there is a light that is coming out because the bacteria are alive and they are bioluminescent. And the areas where the bacteria are dead are dark because that area has no bacteria and no bioluminescence. So let's see on the right, we have these honey samples which are spiked with a fungal toxin called aflatoxin. So you can see these are all the honey samples, and this is the standard of aflatoxin. And you can see exactly which honey samples are contaminated with aflatoxin because of the dark spots. You can also see the bottom of the plate is brighter than usual because this honey has sugar, and the bacteria down here where the sugar has not been separated, they have a double supply of nutrition. So वहां पे bacteria का colony double हो गया. So now you have a brighter area showing the bio-beneficial effects, and you have a dark area showing the toxic effects. So ऐसे करके we use this bio-autography hyphenation to see whether to screen a large number of herbal extracts for toxicity or beneficial effects. Now, seeing a dark zone does not tell you anything about the nature of the toxicity. Is it a heavy metal? Is it a fungal toxin? वो मुझे पता नहीं है. But dark zone दिखा, so I know that there is some toxicity. I have to investigate further using other techniques like HPTLC, MS, yeah, any other technique I have, NMR, and so on. So this is a couple of examples of hyphenation. Now I'd like to take the last 5 minutes that I have to talk about the various herbal analysis that can be done using HPTLC. So first one is the fingerprints followed by bioautographic assays. Now, what is an assay? An assay is something that allows you to see the effects of your extract on a biological system. Now, I'll show you some examples. One is a DPPH assay, which shows you antioxidant activity of your extract. Now, next one is the acetylcholine esterase assay, which shows you anti-Alzheimer's disease activity of your extract. And then the third one here I'm showing you is the alpha and beta glucosidase assay, which shows you anti-diabetic activity of your extract. So ऐसे करके Once we have an extract, we can check its fingerprint for quality control, and we can do bioautographic assays to get the idea of what is the usefulness of my product to the patient or to the consumer. So there are several assays. The other assay also is, one of them is, I'll show you by the end of this slide. Now I would like to show you the hardcore herbal extract ka, what are the applications. One is, this is a example of fingerprinting for purity. Now, you know, saffron, kesar, is the most expensive spice in the world. So there is a lot of economic incentive to adulterate it. So what we do is that we establish a botanical reference material, the fingerprint of that material compared to the adulterants. And you can see here, track number 5 shows you the dyes which are used in nakli kesar. Because when you put that fake kesar in your milk products, you want it to turn yellow. So these are Naphthol yellow S and Sudan red G. So the fingerprint of asli kesar compared to the fingerprint of nakli kesar and the dyes that are put in the nakli kesar, all of it on one plate. Now, I told you that herbal extracts or herbal formulations are many times in India contaminated with allopathic APIs. This is identification of these non-steroidal anti-inflammatories, nimesulide and diclofenac, from Ayurvedic tablets, which is work that we did for a FDA lab of India. This is an opium, afim, which is used as a painkiller directly in the traditional medicine, or by isolating the particular psychoactive alkaloids, which are morphine, codeine, thebaine, papaverine, and nicotine. We are able to separate all of these active metabolites and quantify them as well. Another work we did for a government lab of India. Now, this is artemisinin in Artemisia annua, which won the Nobel Prize for a Chinese scientist. This is artemisinin is a herb which is used for anti-malarial activity in the traditional Chinese medicine. Finally, artemisinin was isolated and identified as the cause of this antimicrobial, anti-malarial activity. They wanted to know what is the best time of the year to harvest artemisinin plant to get the Artemisia plant to get the maximum yield of artemisinin. So they did HPTLC every 2 weeks for 1 year, established that yes, this is the time for best time of the year to harvest the plant. So this is an example of quantification of a secondary metabolite from using an HPTLC system. Now, this is the last slide I will talk about before I move to the end of the presentation. This is Andrographis paniculata, known as Kalmeg in Ayurveda. So you can see I have the image of the fingerprint under 3 kinds of light. I have a multi-wavelength study, which my colleague Dr. Saikat will explain to you if you need to know more. I have a spectrum of each one of those molecules that are present in this fingerprint. And I have scans at multiple wavelengths. So we can get a 360 degree view of the plant using HPTLC. This is alkaloids. Derivatization only showing you derivatization of alkaloid fraction. You know, alkaloids are a very, very important class of compounds. Most of the important compounds in plants are alkaloids, like nicotine in tobacco, caffeine in tea and coffee, and so on. This is Adatoda vasica, known as Adulsa in Ayurveda, which has 2 main active alkaloids, vasicine and vasicinone, which you can see here, which we can isolate, quantify, and we can derivatize only the alkaloid fraction of any plant. In the same way, this is derivatization of the cardinoloid fraction in this Caltropis procera. This is derivatization of phenolics and tannins from St. John's wort and Bibitki. So we can do a multiple class of compound study in Enchrome lab. We do a class of compound study with 11 class of compounds. We can tell you if your herbal extract has more alkaloids or more tannins or saponins or phenolics or flavonoids and so on. So due to paucity of time, I will not go through the rest of my material. I just want to show you one more thing, which will be of interest to academics especially. That is that whenever there is every year there is a HPTLC symposium. And whenever an Indian paper is accepted for that international symposium, Enchrome pays the registration fees of that candidate to go to that symposium. the symposium is on a 3-year cycle. That cycle is once it is in Asia, the next year it is in Europe, the next year it is in USA. So, of course, 2020 May the program was supposed to be in Europe. It was postponed to next year. And what I would like to show you is that Indian scientists are always appreciated at these symposia. Last year, the last time the symposium was held in Bangkok, I am very happy to inform you that an Indian paper won the gold medal at the poster competition. You can see here, this title is unfortunately wrong. This is from Ruya College. She won the gold medal. And the year before that, Dr. Vandana from Sieger Institute, Pune, she won a Euro 2000 scholarship for the most promising researcher. That was at the Berlin HPTLC symposium, which was the year before Bangkok. So now we are gearing up for the next symposium, which is going to be in Ljubljana, Slovakia. Right now, we think it's going to be in July or August of 2021, depending on the corona situation, of course. I would like to encourage all of you to send your papers also to this symposium. I will be there along with the entire ANCROM team. We generally have 4 or 5 people from ANCROM's lab. And we have a group of 40 to 45 Indian scientists generally. So I look forward to having many of you there as well. So with that, I would like to end my presentation. I would like to thank the organizers very much for giving me an opportunity. If any of the work that I showed you is of interest to you, please get in touch with us and we'll be very happy to take it up for you as well. So I would like to thank you all very much. I hope that I've stuck to the allotted time given to me. And with that, I end my presentation. Thank you very much.
Thank you, Dr. Akshay, for the informative session. I am sure everybody has gained something from the session and have enriched their knowledge the HPTLC working. And the session is now open for questions from the participants. So those who want to ask questions may unmute their mics and ask the question, or they may write the questions in the chat box and I'll read those questions.
I think it will be advisable if they write they cannot unmute. So it is better that they write their questions and Mamta madam can maybe conduct the question answer session if Mamta ma'am is free.
Sure, ma'am.
That's okay. Because they cannot unmute. That's why. Let them put their questions in the chat box.
Sure.
Preeti madam, I don't see any questions. So let us not waste time. We shall move on to the second session of Dr. Udin Boktiar.
Thank you.
Let us move towards the second session on the topic, the sustainable collection and processing of plants for pharmacognostical studies, for which we have Dr. Sheikh Bakhtiaruddin as our second speaker. And I would like to again invite Dr. Jankana Rathod to introduce Dr. Uddin and take over the further proceedings of the session. Over to you, Jankana ma'am.
Thank you, Preeti ma'am, once again. Here I am to introduce to our second speaker today, Dr. Uddin. He was born in Kushkuli in the district of Satkhira, Bangladesh. He received his BSc Honours and MSc degree in Botany from the Department of Botany, University of Chittagong, Bangladesh in 1993 and 1995 respectively. Dr. Uddin started his career as a research fellow in NCS Implementation Project, Survey of Flora, Ministry of Environment and Forest, Bangladesh, and began his specialization in the field of plant taxonomy and ethnobotany. In 1997, he received the CICHE Award by the British Council for PhD through Edwin Chicago University Biodiversity Link Project. Dr. Uddin was awarded his PhD from the Department of Plant and Soil Sciences, University of Aberdeen, Scotland, UK in 2002. He is the first in Bangladesh who was awarded the PhD in the field of ethnobotany. Then he left for Canada in 2002 as an immigrant and stayed there for 9 months. In June 2003, He returned and joined the University of Chittagong as a lecturer in botany. In March 2004, he was promoted as assistant professor. Dr. Uddin also enjoyed a postdoctoral fellowship at New York Botanical Garden Herbarium, New York, USA in 2005. In January 2010, he was promoted as associate professor and in March 2015 as professor in the same department and continuing his teaching and research in the field of uh, ethnobotany, pharmacognosy, biodiversity, and conservation. His research activity has resulted 4 books, 1 book chapter, and 51 articles in national and international journals. He is life member of Bangladesh Botanical Society, Biodiversity Research Group of Bangladesh, and member of Bangladesh Association of Plant Taxonomists, Society for Medicinal Plant Research Germany, International Society for Research on Natural Products. He is the Vice President of the Association of Global Traditional Medicines. He presented many research articles in different conferences and symposia at home and on abroad. Bangladesh Ethnobotanical Online Database and Medicinal Plant of Bangladesh are his major contributions in the field of ethnobotany and medicinal plants. May I request sir to please present your lecture. Hand over to you, sir.
Good afternoon, everyone. I'm Professor Boktir from Bangladesh. You have already heard. It's very nice to meet you in this platform because it's a platform we can share our views in this platform. So in this pandemic situation, so I'm going to talk about sustainable collection methods for herbal or plants. Welcome to my presentation. Can you hear me?
Sir, it will be better if you can speak up little bit, little more
Okay.
volume.
Okay, thank you. So my topics on sustainable collection methods for collection and processing plants for pharmacognostic studies. We all know about medicinal plant. A medicinal plant is a plant that is used with intention of maintaining health to be administered for a specific condition or both, whether in modern medicine or in traditional medicine system. So we collect medicinal plants. We collect medicinal plants for herbal preparation as crude drug, local use for medication, for modern medicine. In pharmacognostic study, we collect medicine for the test of bioactivity, antimicrobial assay, phytochemical investigation, and so on. So if we don't collect them systematically, the medicinal plant will be lost from our nature and the biodiversity will be degraded. So to conserve the medicinal plants, we need to collect them in a sustainable way for either pharmacognostic study or in preparation of medicine. So why do we conserve medicinal plants? Because they are the rich source of bioactive compounds. From the morning session, we already know most of the plants, we don't know their phytochemicals or their bioactivity. So they're the rich source of bioactive compounds. They're the important raw materials for drug production in Ayurvedic system, Unani system, homeopathic system, allopathic system, traditional medicine system. They are the important raw materials, source of new drug development. So in pharmacognosy, we use plant materials for search for new drug. They are the commercial source for economic development. We also export medicinal plants from our country, and for local production we also need medicinal plants, and they're the low cost medicine for local use in primary healthcare. So it has been said that 35,000 to 70,000 species of plants have been used at one time or another for medical purposes. So, but the greater number of species is employed in herbal medicine and is used unreferent and semi-processed form. So, we need a lot of medicinal plants throughout the year. In South Asia alone, Ayurveda, Unani, Siddha, Chinese, Amchi, and homeopathic traditional healing approaches make use about 9,000 plant species. The demand for medicinal plant-based raw materials is growing at 15 to 25% internationally, according to World Health Organization estimate. The present demand for medicinal plant is about $14 billion in a year, and it's likely to grow to 5 trillion by 2050. So it's very important to us. The status of traditional medicine in Bangladesh: we have a Unani and Ayurvedic system of medicine. We have Unani and Ayurvedic board. We have 15 government recognized educational institutions about traditional medicine, 10 of them about Unani system, and 5 of them about the Ayurvedic system, and more than 400 big and small manufacturers of herbal medicine in Bangladesh. So they collect medicinal plants from different parts of the country. The map showing some plant materials growing in different corners of Bangladesh. So medicinal plant resources are decreasing. Why? What's the cause for decreasing the medicinal plant? There are some natural causes of decreasing of medicinal plants like flood, landsliding, drought, pollution, disease of plants, and global warming, and so on. Some other causes: inadequate knowledge of medicinal plants. So we don't know about the medicinal plants and we destroy them sometimes. Loss of cultural diversity. Unregulated and overexploitation. It's a very big cause for depletion of medicinal plants. Less supply than demand. Expansion of agricultural cultivation. Overgrazing. Shifting cultivation is one of the major cause for degradation of medicinal plants in our hilly areas. Forest fire, habitat destruction for urbanization, construction of road and highways, monoculture for forest. The main cause for medicinal plant degradation: unregulated and over harvesting, because we don't have the proper knowledge to proper harvesting. Sometimes we do follow destructive method for harvesting of medicinal plants, like we do not leave any plants for regeneration for herbal medicine, over harvesting, or sometimes we kill or uprooted the plant. We picked all the leaves from a plant, so the plant dies. So improper harvest can deplete the population. Medicinal plant harvest can affect other plants and animals. They can affect the plant communities. Medicinal plant harvest can affect population genetics. It can affect the ecosystem. So as a result, the biodiversity cause loss. So this is our focus point: harvesting guideline, the sustainable way how could we collect medicinal plants. So collection of traditional medicine or for the purpose of pharmacognostic study, drugs may be collected from wild or a cultivated source. So it is known that the active constituents of medicinal plants are affected by many factors and may vary during the course of plant growth. Proper time of collection is very often important to obtain a drug of a good quality. The stage at which plant is collected or harvested is therefore very important for maximizing the yield of desired constituents. The best harvesting weather is a clear sunny day, since rains can wash away some of the very constituents we are hoping to gather. When harvesting, we want to be sure to take only what we need for each plant. The first one, we need proper identification. Some plants are toxic and some are outright deadly. It is vital to know exactly what the species is within the genus we are collecting from. Some families of plants do not have common uses across the various species, and some are downright poisonous. And there are some similar looking plant growing in the nature. So we have to make sure which plant we are going to collect. Harvesting guidelines. First of all, we need to choose healthy and well-developed plant materials. Plant that is infested with pathogen or insects should be avoided. This is because byproducts of these organs will alter the ingredient profile and could even be poisonous. Be sure the plants are intended to harvest have not been sprayed with pesticides, herbicides, or fertilizer. Be especially aware around the edges of farm fields, roadsides, or near the industrial activity. Equipment to use. Make sure that all equipment used is clean and free of remnants of previously harvested plants. Keep all containers used during the harvesting clean and free from contamination by previously harvested medicinal plants or other foreign matters. If plastic containers are used, pay particular attention any possible retention of moisture that would lead to the growth of mold. And when the containers are not in use, keep them in dry conditions and in an area that protected from insects, birds, and other pests. So factors affecting collection. Time of the year. Time of collection is very important. Chemical constituents varies in different seasons and time of the day. The plant may contain a substance in winter that is not present in summer, and its amount varies markedly. For example, rhubarb contains no anthraquinone in winter. Instead, it contains anthranol, which in summer are oxidized to anthraquinones. So, if we would look for anthraquinone, we must collect it in summer, not in winter. Colchicum calm is free from bitterness and is devoid of alkaloid colchicine in autumn. Hence, it is used in Austria for food instead of potatoes. Bitterness start in the spring and early summer when it is used as a drug, because colchicine is you can find it in that time in summer, early summer. So if you collect it in autumn, you are not going to get any colchicine. You have to collect it in summer. Time of the day. Some drugs like digitalis contain different amounts of active constituents in different times of the day, being highest in the afternoon. So how can we get this information? Sometimes ethnobotanical information or information from the local user may help to select the time, season of collection of plants. Different parts of the plant should be collected depending on some criteria. This is the general criteria. Leaves and flower parts should be collected during the active reproductive process of the plant, such as from the beginning of the flowering to early fruiting. Flowers should be collected prior to or just about the time of pollination, before the open or just start opening, and collection should be affected in dry weather and towards the middle of the day. So we should avoid collecting plants, flowers, or leaf in the middle of the day. Time of maturity. In case of fruits, fruits should be collected when they are fully mature but unripe. In some cases, when fully ripe, such as amblyferous fruit. Seeds should be collected when they are fully matured and haven't ripened, but before the fruits open. Bark are generally collected at the end of the winter and before the new growing season of vegetative process starts. Root and rhizomes of annual plants should be collected shortly before flowering. Stage of maturity and age. The value of content of active constituents many drugs depends on the stage of maturity and age. Such as conium fruits contain conine when the fruits are mature and unripe. Fully mature but unripe capsule of Papaver somnifera, that means poppy, contain highest amount of morphine. So Xanthonica flowers are rich with xanthonin when unexpanded. When it starts to open, the xanthonin contents decrease. So we have to take care, we have to take in consideration about this matter. So there are, I'm going to propose some guidelines for collecting different parts of plants. In medicinal plants, the medically effective substances are found in the root. Thus, in many cases, the whole
Excuse
plant
me,
gets
sir.
uprooted.
I would like to once again request you to kindly speak up a little bit. आपकी आवाज कम सुनाई दे रही है, तो प्लीज थोड़ा सा पार्टिसिपेंट्स को ठीक रहेगा. Thank you very much.
Okay, I'll try. Okay, can you hear me clear now?
Yes, sir. Much clear, much clear. Thank you.
Okay, thank you. So in many medicinal plants, the medically effective substances are found in the root. Thus, in many cases, the whole plant gets uprooted by gathers and thus can never grow again. If this technique is used within a high number of plants in a short time, the plant will eventually go extinct. In order to ensure sustainable harvest of root materials, dig the root at a considerable distance, at least 30 centimeter from the main stem of taproot. Take it from one side and left it for 2-3 months, and then take the root from another side by digging it. So you do not need to uproot or kill the plants for collection of root. In case of Agal marmoleus or Oroxylum indicum, we do harvest root, so no preparation in, used. The whole tree is cut down and take the roots. So it's a destructive way and lot of waste and degradation of biodiversity. So what could we do? Collect only the mature side of root, tap root undisturbed. So if you collect from the one side and left it for 2-3 months, then the root will grow again, and from after 3 months you can collect from the another side. So the plant will survive. You don't need to destroy or uproot the plant. So you can harvest 2 to 3 times in a year. Tuberous root. The whole branch of root is dig out. Generally, large and medium-sized roots are taken, having a small ones as waste. So what should I do? Collect only large and stout tuberous root, allow further grow. So this is the way of the plant. So this is the way for sustainable collection of root, tuberous root. In case of hemidesmus, we know about this plant, and like this plant, the practice is planting 4-leaf seedlings, raised bed in an emplacement of 1 inch, and its harvesting is too expensive and wastage is high because the root goes very deep in the soil. So we need to, for uprooting the plants or for collecting the root, we it's become expensive. So what can I do? There is a method I have get it from a literature, that 2 meter long, thick, vertically split bamboo stems, then remove nodal portion except at basal node and make holes at lowest nodal portion. Rejoin the 2 bamboo loops using nylon rope. Fill the bamboo stem with pitting mixture, and 2 to 3 rooted seedlings are planted on the top of the bamboo stems and are kept erect on the soil. Harvest the entire root after 2 years by separating the bamboo leaves like this. The bamboo portion, you can collect the root from one side and then cover it, and after 2 months you can collect from another side. In this way, the collection expenses will be lower and no damage and wastage. There is a special method for cultivation in long poly bags. We do collect root from Gloriosa superba, Ulot Jondal. So useful parts: tuberous root and propagation, seeds and roots, cultivated either in soil beds or in mounds, and harvesting too expensive, wastage is high. So what can we do? There is a special method. Half of the polybags are filled with potting mixture. Keep them in 15 centimeter deep pits at a distance of 1 feet. 2 rooted seedlings are planted in each polybag. Bamboo splits are supported to the polybag to the plant. Organic manuring twice at 6 months interval. Harvest after 2 years. Roots are extracted by treating in the polybags. So you can collect it easily and the plant will alive for long time. So no damage and no waste. Herbaceous plant problems in existing plastics, unscientific harvesting of plants like Cedar rhombifolia, Desmodium gangeticum. So what should we do? Collection before seed setting and seed dispersal. Usually we collect it before seed setting and seed dispersal. As a result, the next year we cannot get any plant. So what should we do? Use iron crowbar for losing the soil. Collection after fruiting seasons, because food dispersal will go on, and what will the next year we can get plant from there. And keep a small area untouched for natural regeneration, because if we need for sustainable collection from the same area for many years, so we keep some area untouched for natural regeneration. Bark harvesting. So we can harvest bark for oat June, Kurchi, Holar Hanainte, Disinteca, Termillaria, Urjuna, by cutting down the entire tree or chopping the main stem and branches, removing the entire bark from the existing tree. So what's the sustainable method? So we should remove only 1/3 of the mature bark. Remove only the outer and middle bark, leaving inner bark for regeneration. What's the advantages? Extraction 4 times a year without killing or damaging the tree. So this is the sustainable way. Like this picture, we can collect bark from different parts of the plant, from different side of the plant. So they will degenerate and we can collect it from another part. So we can extract the bark 4 times a year without killing or damaging the whole plant. Fruit harvesting. By cutting down the fruit bearing branches, by harvesting all the fruits from a plant, left nothing for regeneration. So what's the sustainable method? Collect the fruits alone by hand or with hooks. Leave some fruits for regeneration. Avoid damaging the plant. Post-harvest management. Arriving at the place for drying or processing, unload and unpack the plant material as promptly as possible. As soon as we have time to do it, start the appropriate preliminary processing. This could include inspection, elimination of undesirable materials and contaminants, washing, sorting, and cutting. The inspection may include visual inspection. Make sure that there is no untargeted medicinal plants or plant part is included. Visual protection for foreign matter, quality evaluation such as damage, size, color, odor, or possibly medicinal properties. Take care to ensure no foreign matter, weeds, or toxin plants are mixed with the harvested medicinal plants or targeted plant. So in conclusion, we could say that improper harvesting method decrease the quality of traditional herb or the plants we are going to examine in our lab for pharmacognostic study, increase production costs, decline regeneration, loss of traditional herbs, affect other plants and animals, affect plant communities, affect population genetics. So as a result, loss of plant diversity, loss of medicinal plants. So for sustainable use of traditional herbs, we must follow the proper harvesting guideline. So I'm going to introduce about our lab a little bit. We are serving ethnomedicine in different indigenous community of Bangladesh. We have already documented information from 12 indigenous communities of Chittagong and Chittagong Hill Tract, and we have collected about 900 plants. We have 2 databases in the websites, 5 books from our lab, 52 articles in different journals in home and abroad. This is some of our books. And we have published journal with Frontiers, Molecule, Ethnopharmacology, Ethnobiology, Ethnomedicine. And this is our database, Medicinal Plants of Bangladesh. 1479 medicinal plants included in this database. We have another database, Bangladesh Ethnobotany online database. A total of more than 900 plants, now included more than 1,000 plants used for medicine, food, dye, cosmetics, tools and crafts, housemaking, construction, etc. And we have collaboration with different universities in China, Ireland, and Poland. So that's all from me. Thank you very much.
Thank you very much, Dr. Udin. It was really very informative session for all of us. I think there are a few questions. The first question is from Ms. Dr. Malvi Vyas. She has asked about, can you able to provide me some of these medicinal plants that you mentioned in your presentation for further study? She's asking if she can get the plant material from your side.
Yeah. So we have done works with a lot of plants. I can give him just if she give me the email address, I'll provide him all the list of our plants. Okay.
Dr. Malvi, I would like to add into this. Few years ago, that is how I came in contact with Dr. Udin. I was researching one of the plants which was not available in India, and
Yeah.
got it from the MPDPD database website of Bangladesh. And from that, I got contact number of Dr. Udin, and he supplied me the plant material. But to get the plant material from the other country is practically very difficult.
Yeah.
Actually not allowed. Looking to the biodiversity laws of the various countries, it is practically not allowed to get the raw material from the other country. They cannot
Yeah.
pass it on to us, and we cannot pass it on to them. So
Yeah.
it would be a bit difficult for both of you, right, sir?
Yeah. It's very difficult. So because of biodiversity protection law, we can provide. It's very difficult, but we have MOU between two institutions. Then it's easy to share materials.
There is one more question from Dr. Gauri Shankar Juga Prakash. Dr. Gauri Shankar has asked, what about the method of collection of lower plant materials having curative property? What actually is asking about?
Like bryophytes or
Lower plant
pteridophytes.
Yeah.
Yeah, this is the same procedure, but we must care about regeneration of the plants. We don't destroy the whole plants. We have to take care about this, because if you collect pteridophytes from a specific place, you need to leave 2, 3, 4 plants for regeneration. Then you can take all of them. That's not bad, but you have to leave 4, 5, 6 plants for regeneration in that place. For all the lower plants, you have to follow that rules. You have to leave some plants for regeneration in the nature.
So I think Mr. Malvi Prakash, Mr. Gauri Shankar has got his answer from the sir's this thing, whatever he tried to clear about the lower plants and the collection of that. I don't see any other question over here for Dr. Udain, but we do have question for Dr. Akshay Charegaonkar. So I would like Dr. Charegaonkar or his subordinate to answer the question on his behalf. It is one of the question is from Minal Patel. She has asked that what are the challenges for HPTLC methods?
Okay, so the challenges for developing an HPTLC methods are nothing, I would say, because there is a vast literature. If you see the book of Plant Drug and Analysis by Wagoner, that has a lot of being research done into the herbal field. There are several other books which have been compiled over the time period, like TLC by Egonstahl, and for herbarium in India there are many books such as the ICMR, Indian Council of Medical Research, has few publications, the Ayurvedic Pharmacopoeia. So what you need to do is get those methodologies, and you can just transfer them into a format of HPTLC, standardize them, and then it should be easier for developing a method rightwise. The only thing is you have to volume optimize it, and you have to be careful about the conditions in which you are developing a chromatogram. Specifically, that is why humidity control and saturation are being practiced for 20 minutes, as well as the temperature monitor in which you are doing an experiment. These things have to be carefully monitored. For temperature, it gives you slight variation. You will not experience that much of difference in the chromatography, but definitely the humidity and uh, the humidity will be an important aspect which has to be looked in for the variation which you have to control, because that is varying throughout the entire globe, and because of which that implementation of about 33% humidity is there. So these are few things. Apart from them, the volume optimization is just not that difficult, and method development is quite simple. For crude stages, you can do the spot test where you just spot on the samples around 15 spots of the same sample, and then you take clear solvents like methanol, toluene, ethyl acetate, and solvents like that, and you just with the capillary you just allow to overspot these samples and allow it to diffuse. And once the diffusion is being done, at that point of time you get to understand how is the separation in form of a concentric rings. And from that you can optimize method development. You can send us a mail. I'll be discussing more about this with you, and this is a preliminary strategy which we run. Or you can apply samples at multiple spots and run them in different mobile phases and check their like the British pharmacopoeia speaks about 5 different polarity mobile phases, get an understanding from that, and then perform a binary tertiary combination to prepare your mobile phase. So likewise, there is not much of a challenge in developing any of the methodologies. Definitely volatiles are a little bit of difficult samples because they are tend to evaporate even at temperatures of around 40 degrees, which is very difficult to handle. So in that case, GC would be a better alternative. But apart from that, and also high molecular weight molecules, these are there that needs to be analyzed with a plates specifically designed for analyzing high molecular weights. Like you have the reverse phase, then you have the proteomics plates which are available in the market, and on that even protein molecules can be analyzed. So these are the few tips and tricks where you can analyze. So I don't feel anything problem is there. So I hope this answers your question. Next question, please, ma'am.
Yeah, I would like to just shortly say whatever Mr. Malik has said.
Yeah.
With that we have to optimize the conditions. Volume to be spotted of the test solution, mobile phase optimization, humidity is very crucial. Temperature also is crucial. So basic criteria for conducting the entire method is very important, right? So I hope the answer of Mr. Malik has satisfied the question. Next question is from Ms. Jalpa Sanandia. She has asked that I want to know how we use HPTLC in herbal pharmacokinetic study.
Okay. So the herbal pharmacokinetic studies which are there that are revolving around samples from biological, if I'm not mistaken, that is the you do the administration to the in vitro and in vivo conditions, such as you administer it to the rat or mice, and then you take periodical samples, the blood samples from them, and analyze them. So in that case, again, what you have to focus is about the extraction part. That is very simple. Like for any other technique, other chromatographic techniques, you extract them, and once these are being extracted, that can be analyzed. I have just a slide on that. Let me show you for a second. Let me get back to this slide, and I can show it to you. Um, so in that, the same curcumins which are there that have been analyzed, and what they have done is they have taken that serum sample, plasma sample, extracted with the help of the yeah, I got it. So let me show you this example, and let me share my screen for a second. This is one of the presentations given to Naipur Kolkata, and there we had this example. So on your screen, what you will seeing is a live example which has been actually analyzed from a formulation that is having berberine. That is one of the components coming from Berberis aristata. We all know that, and that was studied from the blood plasma. So you can see it's very simple. You have to just apply them and just observe them. So really not a very difficult part. You require a standard for this marker specifically, and now with the IPC coming in with having the 4 marker approach for phyto chemicals, it's simpler on HPTLC as we perceive it. So this is my answer to your question. Hope this answer is your question. Thanks.
Yeah, next question is from Mr. Prashant B.
Okay.
He asked, is there any training offered for the faculty at your organization? The session was amazing, particularly bioluminescence as and MS linked HPTLC. So he has actually praised, appreciated your talk, and he was very happy to be with us. And he has asked about the possibility of training at your institute.
So not only to him, but we offer our trainings to all faculty and such development programs we do conduct. We have a training program actually, but due to COVID conditions, we have restricted those training sessions. We had it actually for a week, but now we have restricted to 1 or 2 days, where 4-hour sessions in a day we conduct, and we provide you with the presentation as well as with the proper live demo of the instrument, with understanding of what process happens how, and also a live sample analysis in front of you. And we are also working on some botanical curriculums which can be shared with most of the pharma colleges and herbal related work areas. And that is a 3 days proper protocol for understanding various aspects, like you have validation, you have quantification, you have bioavailability. There are many aspects associated to it, so we have catered to it to learn the basics of it. So like that we have designed it. So definitely right now we can do it with the help of mutual exchange. You can write to us, and we are right now we have a capacity to take around 30 to 40 people, but right now due to the COVID conditions we have restricted to 3 to 4 people. So 4 people in a batch we can accommodate and provide you with the training at our facility in Mulund. So that is quite possible, and it can be done. Thank you for all your comments and questions, and hope this answers your query. And we are always there to help you out. So anytime you have any queries, to not only to you but the entire researchers, young researchers, as well as delegates, whosoever wants to, dignitaries, anybody has any questions can write to us at lab@ancrum.in, and we'll be happy to help you out. Thank you.
Dr. Malvi Vyas also has expressed the same desire, and she has told that she has written here that she wants to join, or she wants to be part of your institution for analysis of the data on some medicinal plants. So I think Dr. Malik has already said everything about whatever possibilities are there of joining his laboratory for training, or maybe having associated work. So I think other than that, there is no other question for either Mr. Malik and Mr. Udin. So madam, this is where we end the session. I would hand over the mic to Jankana madam, Preeti madam, to give the concluding remarks.
Just stop the screen sharing. Yeah, sorry.
I think madam, ma'am is here.
Yes, ma'am. Thank you, Mamta ma'am, for conducting the question and answer session.
There are multiple devices because of which the echo is coming.
Sorry, there is some disturbance because of some technical problems. Preeti madam is just joining us for the concluding remarks.
I would like to add here before Preetiman concludes that both the sessions were wonderful, and I know that pharmaceutical and botanicals, when we mix together, I think it's a great combination. And especially my students have been writing to me that they have been extremely thoroughly enjoyed both the lectures, and they have been initiated into more analysis and thought-provoking research. Thank you very much. I think Preetiman will join us soon.
Yes, madam. I also would like to share my opinion. It was really nice to have both the speakers with us today. Both of them gave us very nice information regarding the topics they covered. And I hope not only pharmacy students, but botany students also must have been benefited through the talk, because medicinal plants are of prime importance these days. And as such, if we see the drug discovery, it starts from the nature
Always a scientist who is there in the drug discovery field goes to nature to find a new molecule. And there are n number of possibilities of getting success when you go to nature, right? So,
ma'am.
I convey my thanks to both the speakers.
Yes.
Over my mic to Preeti Madam.
Madam,
Madam, she is joining. Sorry for the inconvenience.
That's okay, ma'am. Go ahead, Preetiben.
Yeah, am I audible, ma'am?
Yes, ma'am, thoroughly.
Sorry for the technical issues. So ma'am has thanked both the speakers. So I would like to conclude this event of today. Before concluding, there is an announcement for the participants that the link for the feedback form will be given at the end of the event tomorrow. E-certificates will be given to only those participants who would be attending all the sessions and would fill up the feedback form too. With this, we have successfully completed the first day of our international conference. We will meet again tomorrow for the next session at 11 a.m. sharp. So thank you all for being with us today. Thank you for all the organizations. Thanks a lot and see you tomorrow. Bye-bye.
Thank you, everyone.